Texas Lutheran University.
It's my pleasure to welcome Dr. George Yatskievych. He got his bachelor's and master's degree at the University of Arizona at Tucson, and then his PhD at Indiana University. He spent 28 years as the curator at the Missouri Botanical Garden, doing research mainly on the flora of Missouri before moving to Austin last November. So he's been here for a little while now, almost a full year, where he became the curator at the Plant Resources Center at the UT Herbarium. So let's welcome Dr. Yatskievych.
Well, thank you. Just to clarify one thing, if you sit on that side of the room, you can still get into the picture on the camera. So I figured that's why everybody was sitting over here. Anyway, I am a botanist. That's my addiction. And in particular, I'm a plant taxonomist, which some people say is the worst form of botany. What I want to do today is talk about some work that I started a number of years ago when I was in Missouri. Doesn't really concern Missouri as a unit, it's Mexico. But like so many of my projects, it never really finishes for good, and so it's still carrying on in various ways. And in the process, what I want you to sort of keep in the back of your mind is the idea that this is sort of a template for how plant exploration and basic plant taxonomy work at a professional level, and it sort of gets expanded or contracted depending upon the kind of project, but it's all variations on the same theme. So what we want to talk about today is parasitic plants. And I have to admit that when I first got interested in botany as a sophomore at the University of Arizona, very quickly I developed an affinity for the oddball plants, the ones that didn't follow the rules, that didn't do things in their life histories the way that normal plants did. So parasitic plants are the ones that are the botanical freeloaders. They get their food and water from other sources than photosynthesis. And you know, we learn about photosynthesis in introductory biology courses, but that's not the way that all plants work. And there are gradations of parasitism, so that some of these plants look very much like regular plants, and until you dig them up and see that they're connected to hosts, you never know that they're parasites. And then others have gone to the other extreme where they don't even look like plants. These look like the ends of little baseball bats sticking out of the sand, but that's a plant with flowers. There are two broad classes of what we call heterotrophic plants. The one is the parasites that have direct root attachments to a host plant, either in the roots or in some cases, like daughters, they're on the stems and leaves, various parts of the plant this parasite can latch into, and it forms a connection with the vascular tissue and funnels out the water and the food from the host plant. The other group, which has this horrible long name, mycoheterotrophic plants, are the plants that actually parasitize fungi that are in the soil. And those same fungi are the same fungi that form mycorrhizal attachments with tree roots. So in fact, it's actually been shown with radioactive sugars that nutrients from the tree can pass down into the soil-borne fungus and then wind up in this parasite. So it's like a secondary parasite. And these two basic modes are the way that plants have set aside photosynthesis and decided to get their energy from other sources. And there are, as I'll mention again later, for the parasites, approximately 30 different plant families scattered throughout the flowering plants, and even one gymnosperm, that do this parasitism, and another set of families that do this mycoheterotrophic thing. So it's actually a fairly widespread phenomenon that has developed over and over and over again in plants. So this particular story goes back to 1985. And there's a fellow named Wade Thomas who is a curator at the New York Botanical Garden, who is very well known in our small little society of botanists for being an explorer for plants. And in 1985, he was in southern Mexico on a trip looking for some plants for a research project. And one night when they made camp, he noticed this odd little plant sticking out of the ground, pulled it up without thinking about it too much, made an herbarium specimen by drying and flattening it, and then when he got back, couldn't figure out what it was. He took a picture of the plant because these plants that don't have chlorophyll oftentimes discolor and distort, and so they're hard to figure out what's going on with the plant to characterize it. So he had this nice image of the plant, but even that was pretty otherworldly, really didn't look much like a plant. He couldn't figure it out on his own using the manuals and taxonomic works of the time, so he did send the plant to an expert on the Orobanchaceae, which is the principal family of root parasites in North America. And this fellow, the late Larry Heckard, wrote that it's not known to him, and he was the expert, and it might not even be a parasitic plant, but it might be one of these myco-heterotrophic plants in this monotropoid group of Ericaceae. So we weren't even sure if this plant was a primary or secondary parasite because we didn't have the connection to anything at the bottom because it broke off. He then later also sent the plant to a guy in Vienna who was another expert who didn't know what it was either. And so this specimen accumulates all these little notes that basically have question marks all over them. Pretty weird looking for a plant. More recently, Thomas found that there was a group of us that are working on the plant family Orobanchaceae, at least some of the genera, for the Flora of North America project. Flora of North America project is a project to do an encyclopedia of all the plants growing in the US, Canada, and Greenland, and it's going to be a 30-volume set, kind of like the Encyclopedia Britannica, and so far 19 of those volumes have come out, and we're still working on some of the rest of the families, and it involves hundreds of scientists from all over the world that are contributing their specialty groups. At any rate, he found out we were working on these, so he asked if we would look into it, and I got interested enough that I developed a project around it and developed a little bit of funding that allowed me to do the research. I went to Mexico twice, looking specifically into this project, once in 2006 and one in 2007, a month later. I had the privilege of going with this fellow, Jose Luis Contreras, who as a very young man had been the sort of local guide for Thomas in the mountains of Mexico on that trip in 1985. And so he had some vague idea of where they had gotten this thing. The second trip I went with a colleague and friend of mine, Mark Olson, who had been a graduate student in St. Louis earlier, and he then took his master's student, Laura Trejo, who was interested in finding samples. Her particular project was the wild origins of the cultivated poinsettia plants. And so we saw some of those along the way, and I'll show those. And the reason we had to go twice was we didn't get all the information we needed the first trip, because the first trip, when we finally found the plants, they were just coming into flower, and we needed to see the fruits and the later stages in the life history as well. So to orient you, Mexico City is here, and this is the main highway that goes down to Acapulco. And at some point, you branch off to the west, and the map, the Mexican road map, makes this look like a superhighway, but in fact, for most of its length, it's sort of a logging road. That often happens in countries where they sort of have hopeful ideas of what these roads are going to turn into. What's interesting about this road, and I'll show you, is that it starts out at a low elevation in a very dry habitat, works its way up, and eventually at the highest elevations is this really neat cloud forest with fir trees and mosses hanging from the trees and very dense on the ground. And our plants were somewhere between those two extremes, and so the challenge was to find it. And it took us quite a long time to actually find the plants. So you start out in this Rio Balsas, the Cañón de Zopilote, that's many of you know Spanish for buzzard. It's a very well-known collection historically where a lot of plants new to science were discovered in the 1800s. And it's a very dry vegetation with very widely spaced plants. The vegetation type is known as thorn scrub. This is sort of near where you turn off the main highway. And there are interesting plants there. This manihot, for example, is a wild relative of the manioc plant that's a very important food plant for many people. And this is a much smaller version with fewer flowers that's just endemic to that region. The Lysoloma is a species of legume that's a tree, and this particular species, again, is in that region and not very many other places. So pretty plants and neat plants that sort of distract us from our main purpose for being there, which is good because if you don't find what you're looking for, you need to take something home with you. As you go up in the mountains, the thorn scrub gets denser. You run into what I call road hazards that you aren't expecting, like a big flock of goats. It took us about 20 minutes to get past them. And then eventually you go through these different vegetation zones as you gain elevation. And where we are is on the inland slope of the Sierra Madre. So the coastal part has a wetter slope that goes up to the top, and then there's a drier side on the inland slope because all of the moisture gets exhausted when it comes up that elevation on the coastal slope. And the places that we particularly went through to get to the area where we needed to be was not the highest elevation, but from the tropical deciduous woodland through the oak forest and then eventually through a pine oak forest. And there are all sorts of neat plants. As a botanist, it's fun to go to a new place because you learn lots of new things. There are several species of mountain mahogany, Cercocarpus in Texas, and this was one that I'd never seen before. Lots of little dwarf palms. This is actually a genus that we don't have in Texas very much. Morning glories, which here twine on people's fences, grow into trees down there. So some of the plants that I could recognize had very different growth forms. There were also these weird areas where you had these highly dissected limestone ridges, where if you took a wrong step, instead of stepping on the ground, you'd find there was a big hole. And it was very sharp where the edges of the rock were, so it was very actually dangerous to walk around there. But some of the really neat plants were there. Cestrum is a member of the potato and tomato family, and the Bouvardia is a member of the coffee family. And again, as you're down there, you don't just pay attention to the plants, but you see that they have all these neat insects and birds and other things that interact with the plants. So if you're a naturalist, you get more than just the plants out of the experience. Laura is here with Euphorbia cornastra. It's a very rare and endemic. That is, it's just in that part of Mexico, in that one little area with the dissected limestone. And this is a wild relative of the poinsettia that grows very, very big, and it's a very strong shrubby perennial. There are also neat ferns. My other specialty is studying certain groups of ferns, and so I just thought I'd show one of the prettier ones where the underside is covered with this white powdery material that helps retard water loss and keeps insects from chewing on them, and then the spores are the brown areas along the edges of the leaf. When you get up into the oak and pine area, it becomes very monotonous. Just acres and acres and acres of oaks and pine trees with not very much underneath them. And I should point out that this is one of the areas of Mexico that has the highest rates of timber removal. They're really cutting down these forests very quickly. Once they cut them down, they usually burn the area so that the trees don't come back, and then they turn them into pastures or farmland or whatever they can use to make a living off of, because it's also an economically very depressed area. I should also mention that we can no longer really go to this area now because the drug cartels have set up shop in this part of Mexico. And in 2011, when I attempted to go back to this area, the federal police turned us back because they said that there was some active gunfire going on up the road. And so the study that I did then probably wouldn't be possible now. We just lucked out on getting there before the political and economic climate changed to the point where it became impossible to do the work. So in amongst this sort of sea of oaks and pines, you do get little plants that come up like this wild violet, which is endemic to the region. But more importantly, every once in a while, you get things that break up the vegetation. And you wouldn't notice it looking at Google Maps. The area where we're operating is there, and aside from the road and the power line cut that's over to the left of it, there's really not much of any development that shows up there in the mountains. It's really very rugged. But what happens is you get these little drainages that cross the road. Here's one looking just from the road out, and they are steep-walled and narrow and very tall, and they have a very different vegetation in them. And you get into this drainage, and it's just abrupt because the moisture is trapped in these drainages, and the trees overarch on either side, so you get very tropical plants, a very tropical semi-deciduous forest taking place in these drainages. And you get lots more species, and you get ones that only grow in these little ravines and nowhere on the open slopes. And that's where it turns out our plant grows. And it took us several days of looking before we stumbled on it the first time. Being botanists, we made lists of all these plants that we encountered at these sites so that we could later relate the vegetation specifically of those ravines back to our plant. And there it is. And until you're actually looking right at the plant, you don't really see it because it blends in with all the leaf litter and it kind of looks like a little pine cone sitting on the ground. So one of the reasons that people hadn't experienced this plant since 1985 was because it's so hard to see. And it really does look very unplant-like. Here's some more pictures of it a little bit closer up, and even when you get closer, it really doesn't stand out. And most of the plant is under the ground. It's only the flowers and fruits that stick up on top. It's like a little hand grenade sitting on the soil. And there it is even closer. When you actually get fairly close to it, it's actually pretty. It has subtle colors, but you don't notice those until you're just literally nose to it. And there am I, digging up one of these things. We wanted to find out what its attachment was, whether it was attached to fungi in the soil or whether it was attached directly to the roots of a host plant, because then we could address this issue of which major plant group it was a member of. And the stems are somewhat distorted. You know, they have places where they get crooked, and that's because the soils are so rocky, and as the stems come up through the ground, they can't just grow up, they have to grow around the rocks. So this is the attachment point, and what this is a section of root from a tree. We only actually cut one of these because these plants are so rare that we didn't want to damage the population too much. Plus, it's not really great for the tree to have a 6-inch root cut off of it. But we wanted to do this at least once to demonstrate that there was a connection, and we wanted to show that this is a perennial. So these are the old parts of the plant that died back, and here's new ones coming up. And so in this root, there's a connection to the xylem and the phloem, the vascular tissue, and then this plant forms this sort of plate-like mass on the outside, and then it only sends up these stems when it wants to flower and fruit. So if you know about regular plants and you think about the plants that we interact with walking around campus, this is a very different plant indeed. One of the other problems then was, what is the host? We were ill-equipped the first year because the host is this tree about 75 feet tall with all of the branches and flowers and leaves all the way up there, way above where we could collect it. So we had to wait for a year and come back with better equipment in order to actually harvest a sample with a pole pruner, a big long pole with a pruner on the end of it, so that we could finally figure out what the host was, because just from the trunk we weren't able to identify it. Although it was interesting that the roots and the bark, if you scratched them, had this sort of pungent smell. And that's characteristic of certain what we call primitive angiosperm families. So we had some ideas, but we really didn't know. Well, the host, as we discovered the second time around, is this plant called Heteosmum in a family called the Chloranthaceae. Chloranthaceae are a tropical family that does not occur in Texas. It doesn't get up this far. But as you can see from the map, the host plant is actually fairly widely distributed. This is the tropics line, and from here in central Mexico, it goes all the way down to nearly South America. And in fact, at the time that this was done in 1988, nobody had collected the host plant in the area where we wanted it, which is right about here. And so we're filling in dots for the host as well. The point is, I've gone back to Mexico, to some of these other regions, and done searches in populations of the host plant and never found this parasite again. So this parasite really is stuck in this one little area. From the standpoint of a species, that's probably not the way it's always been. Species at some point have to be more common in order to be successful, or else they die out. And probably what's happening with this species is a combination of time, inability to disperse out of these narrow, steep-walled ravines, and the fact that we're destroying its available habitats very quickly. This plant is clearly on its way out. And in many ways, I feel sort of privileged that we're able to find and describe this plant as new to science before it disappeared from the earth altogether. You know, you figure there are probably, it's been estimated, about 75,000 species of plants out there in the world somewhere that we don't know about yet, waiting to be discovered. And you have to wonder how many of these plants are going to disappear before anybody ever takes notice of them. In characterizing this species, we did a number of things to look at different parts of the plant. many of these parasites have a reduced vegetative structure. That is, in many of these groups of parasites, the leaves are reduced to these little scales. And the vascular tissue becomes very simple, surrounded by these thick succulent layers that are basically starch and water, at least cells containing them. So there are not very many characters in the vegetative parts of the plant that you can use to differentiate this plant from others that it might be confused with. But in the inflorescence and floral morphology, there are all sorts of characteristics that, as a botanist, you can delineate. And so we were interested in categorizing these, illustrating them both with photographs and with a line drawing, so that when we described this plant as new to science, people would know what it looked like. And that's part of the job of being a plant taxonomist. We also, as I said, I had to go back the second year in order to get the fruits. We also looked at the fruit and seed morphology. And these are what the seeds look like. These are the fresh seeds under a microscope, and this is an SEM view. These seeds have these big cells in the outer walls. It's very characteristic of this plant family, Orobanchaceae. And at maturity, the outer walls of these big balloon-like cells pop off, so it's a network. And what this does is it allows wind and water to catch the seeds more effectively. Particularly water, what it does is it basically forms little air bubbles so that these things can percolate through the soil when it rains. We looked for things that were unique. If we assume that this plant is Orobanchaceae, we looked for things that were unique within the family. And we found a number of characters in the stamens and in the cross-section of the ovary, and I'll go through these in more detail. But the point behind this slide is that this, even by standards of this family, was a very odd and unusual plant. We also looked at the pollen, and this turned out to be very weird. Most families have pollen grains that have pores. The pores are so that the pollen tube can escape and get down to pollinate the ovule and turn into a seed. Most of them have what we call equatorial pores. That is a ring of pores, 3 or more, around the middle of the pollen grain. But this one had 6. It had 4 around the middle and 1 at the top and 1 at the bottom. And there's a fancy word because scientists love to invent fancy words to describe these things. Pantohexaporate. 6, and panto meaning that it's all over the place as opposed to in just one area. So this pollen is unique. Pantoporate pollen shows up in a variety of plant families, but it's not very common anywhere, and it's nowhere to be found in the Orobanchaceae. So we can summarize. We knew that this plant was probably going to be Orobanchaceae because it had a direct attachment to another plant root as opposed to being attached to the fungi. But it also had these characters of bilateral symmetry of the flowers, which we call zygomorphic, whereas the Ericaceae and the other things have flowers that are radially symmetrical. It also had stamens fewer than the number of perianth parts, that is the petals and sepals, whereas the Ericaceae have as many as or twice as many as. It had this elongate style with this discoid stigma that was pinched in the middle, and that's not the characteristic of the Ericaceae. And it had these small seeds with that reticulate, that is that network of outer layer cells, which isn't found in the Ericaceae. So we had not just this hunch, but we had actual empirical evidence that this thing was a member of one family and not the other. And it wasn't just the habit. Within the Orobanchaceae, though, we found that it had all these characters that no other members of that family possess. The free sepals instead of the sepals fused into a cup. Filaments free from the corolla as opposed to being fused to the inside of the petals. Pubescence of these weird bladder-like hairs, these orange hairs that are on the outside of the flowers. Anthers in which the stamens have the anthers that are partially embedded in the filament. They're actually sunken in as opposed to being a separate structure, which affects how insects can gather pollen from these plants. Ribbed and warty ovaries and fruits, which are not found anywhere else in that family. 5 placentae, placentae being the places where the seeds are attached to the inside of the fruit, or the ovules inside the ovary, as opposed to 2 or 3 elsewhere in the family. And this weird pollen type that I talked about with the pores at top and bottom and 4 around. So our conclusion based just on the morphology was that, yes, this probably was a member of the Orobanchaceae, this particular plant family, but it was so distinct from any of the other members in that family that we had to call it a new genus, not just a new species, but a new genus. And several years ago, we published this in the genus Novon, and we called it Erematilla mexicana, which is Latin for the little hermit of Mexico, because we're poets at heart. Now, as a taxonomist, it's great to be able to set something aside and say why it's unique, but that's only half the job. Taxonomists are also interested in figuring out what the relationships are between plants. And so the other half of this becomes more difficult, because on the one hand, vegetatively, these parasites have very reduced structures, so there's not very much to go on in terms of comparing morphologies. And on the other hand, this plant had such weird floral morphology, so different from any other members of the family, that there's really no basis for the comparison. So for that kind of thing, you have to turn to a different set of characters. You go into the laboratory, you extract the DNA, you look for sequences of various marker regions within the DNA, and you actually compare the genetic code of these organisms in order to get a better idea of the phylogenetic relationships. Within Texas, by the way, there are several species of these Orobanchaceae that have also lost all their chlorophyll. There is Canophilus, which has the non-PC name of squaw root. And there is Epiphagus virginiana, which is called beech drops because it's always parasitic on the roots of beech trees. Canophilus is always on the roots of oak trees. And then there are several groups of this genus Orobanchae, the broom rapes, some of which are native and two of which are non-native crop weeds that have come into the state as contaminants on various crop seeds. And this one Orobanchae minor started out sort of somewhere in the Houston region and migrated up, hit I-35 and is now even in the Austin area. And these are actually considered noxious weeds by the federal government, and their import is actually, if somebody could figure out how this got in, somebody would get a fine for not cleaning up their crop seeds well enough. There are only really two other genera in North America of these chlorophyll-less Orobanchaceae, and I'll explain that in a minute. But Copsiopsis has two species that are on the western seaboard in Oregon, California, Washington, and British Columbia. And then Bosniakia rossica is in northwestern Canada and Alaska, and it actually goes around the Pacific Rim into China. And all of these plants, when you get to looking at them, are all weird by plant standards, and that's sort of one of the recognition characters for these things that have lost their chlorophyll. In the traditional sense, back in the 1890s, the Orobanchaceae were considered to have about 14 genera and about a total of a little over 150 species. And they had this worldwide distribution, and they all had this character where they didn't have any chlorophyll. They were parasites that depended 100% on their hosts. By contrast, there was this related family, Scrophulariaceae, and each one of these denotes a genus, but I'm not expecting you to read them, so it won't be on the quiz afterward. But the idea of Scrophulariaceae was that it was this huge amalgamation, 177 genera and more than 3,000 species, and several of these groups not only had members that were autotrophic, that is, just used photosynthesis, but they had members that had varying degrees of parasitism. So hemiparasites, like the Indian paintbrushes, we have one species that's very common in the spring in our area, they have these connections, often very fine connections, to the roots of their host plants, but they still have chlorophyll, so they still make part of their sugars themselves, and they only get some of their water and some of their sugars from the host plant. And then you have this gradual sequence until you have a few members of the traditional Scrophulariaceae that actually have lost all their chlorophyll. And there was this problem for students and researchers and others of how to tell these two families apart because they kind of graded into one another. And they started out being described at a time when people only knew about 6,000 plants worldwide as opposed to the 380,000 that we know now. And so at the time they were first described as separate families, they didn't have all these intervening genera. So this had become a major question in plant phylogenetic research about what were the real true limits. And again, DNA was the answer because the plants morphologically weren't telling us the whole story. And I don't want to tell you that all of this has been figured out. This is all stuff that people are continuing to refine with additional sampling of species from around the world, as well as new genetic markers and new technologies that have been developed. Our whole idea of how we go about sequencing DNA has changed over the last 10 years from small target sequences to taking the entire genome and cutting it up and looking at very large data sets, as our ability to compute these things as well as our ability to generate the data have improved. These are base pairs, which are nicely color-coded, and you can see that some of these plants have a deletion and others don't. And so these are the kinds of information that would say, okay, these samples are probably from species that are more closely related. So the idea is that we're looking for various small differences in the genetic code, mutations probably brought upon by ultraviolet light or other mutagens in the environment naturally that over time accumulate in plants. They're neutral. They don't affect the ability of the plant to do its life history, and so they're considered good markers to show how plants diverge over time from one another. Several of these studies started taking this big family of Scrophulariaceae and showing that the members of that family really were not their own closest relatives. And so, for example, this larger study, the members of the Scrophulariaceae are the ones in blue, and you can see that many of them have relatives from other families that are more closely related by this data set than the things that were related to one another in a traditional sense. So there was this, what they call the disintegration of that family. And one group of those, up here in the top, based on this limited sampling, remember there were like 3,000 species involved, and this is a very small sample, was shown to be more closely related to the Orobanchaceae in purple than to the rest of the Scrophulariaceae. And there's a close-up of that part. And so the idea was that there was this incidence where parasitism arose within this group, and some of these things had been called Orobanchaceae and some of them had been called Scrophulariaceae, but they were all part of this parasitic lineage. And parasitism was nowhere else to be found in any of this part of the plant kingdom than in this one lineage. There were subsequent studies that added species to the mix and found basically the same result with more resolution. And again, the idea was that as we went along, we were able to delineate that there was only one incidence of the development of parasitism, and all these other things that were related to one another all went back to some ancestor that was the first parasite. The ones in purple here are the ones that lost their chlorophyll altogether. And so what you see is that there are a lot of these hemiparasites, and in 3 different instances, there are clusters of species that have lost their chlorophyll altogether and become holoparasites. And I'm giving you background because eventually we're going to stick our plant in here and see what it's related to. And finally, there was a big study with even more species that basically showed the same thing. So the total number of Orobanchaceae, when this did this reclassification, went up from just 14 to 17 genera, all the way up to nearly 100 genera, and the total number of species from the traditional went up to over 1,700 species. So the Orobanchaceae in this new circumscription is one of the largest and most taxonomically diverse groups of parasitic plants anywhere out of these 30 or so families that are scattered throughout the flowering plants. Oh, and I should mention, if anybody gets interested in this, the best source for information on the web is Dan Nikrin's website, the Parasitic Plant Connection, which has all manner of information on anything you might want to know about parasitic plants, including these ones that are the myco-heterotrophic plants, and all sorts of cool photographs and stuff that's just available to download. And it's a great resource for teachers who want to teach about parasitic plants too, because it's not just lists of plants, but it's all sorts of things about their natural history. Not that I expect everybody to rush right out and do that. So first off, we wanted to know, do the molecular data also support the placement of Erematilla in that plant family? And what is it most closely related to? Is it most closely related to one of these other clusters of things that have lost their chlorophyll, or is it an independent origin of that loss? And I worked with 2 specialists in this. Gerald Schneeweiss from the University of Vienna is one of the people who's doing those big studies, and the other person is Sarah Matthews, who at the time was at Harvard University, but has since gone to Australia because she likes to move around. She was in Missouri before she was in Harvard. She's sort of a free spirit when it comes to travel. This part has not been published yet, so this is the part that we're still working on finalizing. What we found was that our plant came up within the Orobanchaceae, within this one instance of parasitism that includes all of the North American material along with a few Old World things. So yes, our study did support that, and in fact, it comes out as sister to this group of Bosniaceae that are now called Copsiopsis that I showed you earlier that are along the west coast, basically. So our plants, being near the west coast of Mexico, are just sort of an extension biogeographically of that, and who knows what might have been in the intervening region that hasn't been discovered yet or has gone extinct. And the only reason that I put this up again is to show you that these things aren't just variations on a theme, but they're actually using different hosts, and so they have different life histories. And so the two species of Copsiopsis are parasitic on various members, shrubby members of the Ericaceae. Arbutus is a genus we have in Texas, and the rest of these are mostly things that are out west. Arematilla is on this Chloranthaceae, which is a very different host. Conophilus, the two species, are on oaks, Quercus. Epiphagus is on beech trees, and then we have these other species that also have different hosts. So these things diversified some time ago in different places of the world and are doing different things with different hosts. And the bottom line is that there's tremendous opportunities for studying various aspects of the life histories of these plants that, at least with my Arematilla, we didn't get a chance to study. So for example, we have these weird gaping flowers. What kind of insects are actually pollinating these things? Good question. Let's go down and find out. And other things about what's limiting the distribution of this plant would be interesting. Why isn't it elsewhere in the range of the host? We don't have any empirical data to support one hypothesis or another. So there's lots of things that we could still be doing with this plant. So to conclude, the molecular data support the inclusion of Arematilla in this particular plant family, Orobanchaceae. It's derived from other holoparasitic species, that is this group of North American species, primarily North American, and it's sister to this wholly Western North American Copsiopsis, which makes sense biogeographically because things like Epiphagus are primarily in the eastern US, and they only make it as far as eastern Texas and northern Mexico. As I said, there's still lots of questions, and what you find in biology in general, if you go on to graduate school and you start doing research toward a thesis or dissertation, one of the most troubling things to any student is that you never quite feel like you're done at the point when you have to be finished in order to get your degree. And I feel like that with every project that I ever start, and it's a wonderful thing because it always gives me things to come back to and explore more in the future. And I think I've said enough. Thank you. Yes.
So I noticed, like, on your different
Trees.
families that you, like, did the DNA barcoding for, you use different barcodes for different ones. How did you kind of decide? Was it just, like, technology increased and so you realized you needed to use better ones, or?
Well, it becomes very critical to select the right markers for these sequences. In other flowering plants, many of the marker sequences that are used are genes that are ubiquitous in the sense that they're involved in photosynthesis. In other words, you want to have these markers that all plants are doing the same thing, not just something that's unique to a particular group of plants. Well, in this group of plants, and in most other parasites that have lost their chlorophyll, there's no reason to have photosynthesis. So there's no reason to maintain the DNA and the genes that do this. So in other words, there's no going back. Once you're a parasite, you tend to lose that part of your genome and you can never reclaim it. So in choosing things, we choose primarily sequences that are either part of the metabolic portion of the plant life history. In other words, the part where they're taking sugars and turning them into energy, or things that are what we call spacer regions that have no defined transcription areas that actually have active genes that make a product. And these neutral regions essentially just, nobody really knows exactly what some of them do because they don't have this known function. But the other thing is you want to choose regions that have a level of variation that's not so excessive that you can't study it. In other words, you have to have a conservative enough DNA that you have a framework for doing these comparisons, but you want to have enough variation that there is variation. So there are papers that have recommendations for which markers you should use. And these are not necessarily barcodes because they may work in Orobanchaceae and related families, but they might not work in oak trees. So there are different markers for different parts of the plant families. And these were ones that my co-authors and their predecessors had used successfully. And so we didn't want to have to reinvent the wheel and try out a bunch of things. Because when you start a study like this from scratch, oftentimes you don't know what the right markers are. And you waste a lot of time with genes that basically turn out to be invariant across your species group. So there are no mutations to see at all. Or there's so many piled on one another that you can't analyze the level of variation present. So we got lucky in having this data set that was available ahead of time, and then we could make use of that. We weren't being innovative at all.
Are these parasitic plants at all harmful to their host?
We don't have any evidence that any of the native North American parasites do much to their hosts. And in fact, in another group that I was studying, the Lenoaceae, which are the little baseball bats I showed at first, which grow in these sand dune areas, you could take the plant, excavate the whole system, and which was done in the 1920s, and the parasite outweighed the host by a factor of 22 times. And yet it had no visible effect on the host, despite growing in the driest part of North America where rainfall is less than 3 inches a year, and these parasites have these thick, succulent, water-filled stems. There's even some indication maybe that the host is able to siphon some of that moisture back into its roots during really, really dry parts, but nobody's really studied that in any detail. That's just speculation.
So it might be symbiotic, kind of?
It could very well be, but nobody really knows. In all cases, it's stupid for a parasite to kill its host because you kill yourself in the process. So these parasites may weaken their hosts. What happens with the crop plants is that something that can latch on to a new and unique host doesn't have that time to build up that sort of tension, that sort of equilibrium. And so we get these high reductions in the yield of crop plants simply because these parasites weren't actually designed originally in the wild to parasitize these cultivated plants, but when they come in contact with them, if they're happy, they're very, very happy indeed. And so that's where we get these weeds. And there are weeds, particularly in tropical crops, that just wipe out entire crops. And these are usually annuals because many of our crop plants, particularly the grains, are annuals. And so this is a big deal for the people who depend on those for food and for the food for their livestock. And this is why the US government tries to keep these things out, because if they get into tomato fields or cornfields or horrible tobacco fields, that would be even worse, then they can have real impact on things. There was a parasitic Orobanchaceae that showed up last year in a tomato field in the Central Valley of California. And the government quarantined the entire square mile and basically nuked every field within there to sterilize the soil so that this thing did not become more widespread. Now, history shows that none of these attempts to sort of eradicate a parasite ever work. But it makes people feel good to try, I think.
Other questions?
For the molecular work, where did you get the material to use for the molecular work and then to do the analysis that you had?
With the new parasite, the Erematilla, we actually collected extra material in silica gel while we were in the field. Silica gel is the stuff that people use to desiccate things. It's in the little bags when you unpack your electronics and it says, "Do not eat this." People also use it to make dried flower arrangements and various other things. What it does is it dries the contents very, very quickly so that the cellular contents are preserved without lysing and getting all over the place. One of the problems with just drying a plant is that the vacuoles and other places where the cells store tannins and other things that bind proteins and degrade DNA, as they dry slowly, these things pop, and so it ruins the tissue for future study potentially. But by putting things in silica gel in the field, that's sort of a good way of preserving small parts of the tissue so that you can use it for future work, not just DNA, but for phytochemical work. If you wanted to extract particular classes of compounds, you want to stabilize the tissue. Zoologists have the option of putting things into liquid fixatives, and they've developed techniques for working with their animal tissues that have been preserved in formalin, acetic acid, and alcohol mixtures of various sorts. With plants, that tends not to work either. So with plants, we have to dry the tissue before we can work with it.
I have a question. Is it known how you go from a seed to a connection with the plant? Seems like the seeds are fairly small, right? And so how do they get enough tissue to find a host?
Well, the first question is, how does the seed get down to where the roots are? And the answer there is that probably over time, these seeds are long-lived enough that rainwater washes them down through the soil and they percolate down. The root that I excavated was like that far under the ground, so that seed had to go a long way to get to that root. The studies that have been done on some of the other species of Orobanchaceae indicate that when seeds are sitting in the soil, they won't germinate until there's a host root that has a tip near them. Host roots will exude compounds that cause the seed to be stimulated to germinate. And these are compounds that are related to gibberellic acids and other growth factors. They've actually been analyzed in the lab phytochemically, and there are these complex things that are related to gibberellins and auxins and other growth factors. And the host root has to be in close proximity to the seed in order for there to be enough of those factors to stimulate the seed. So it's a very inefficient process. And these seeds, some of the crop parasites, the seeds can last in the soil 75 years or more. So once a plant gets into these fields and is able to set seed, that field will potentially have new parasites in it for ever after for the entire life that you might have as a farmer. So it can become very difficult. But on the other hand, unless you have a very dense crop of suitable hosts, you never have very many roots that come into contact with the seeds. So it's only in artificial situations like crop fields where you have this dense zone of roots at a particular level that you get a lot of this large populations of these things coming up. Elsewhere, it's like one here and one here and one here. Except maybe sometimes in grasslands. You know that our crop of Indian paintbrushes in the spring, it's not just one here, one here, one here. And it's because they do parasitize the grasses, and so there's lots of grasses with interlocking roots. If you ever try to dig up an Indian paintbrush, by the way, the connections between the parasite and the roots are so fine that they almost always break when you dig them up, which is why for a long time nobody knew that they were even parasitic.
Can I ask another question? From a practical from a practical standpoint, how did you then find out that that was the host plant? I mean, you said you dug down this deep or whatever it was, and how did you then know it was this member of the Chloranthaceae?
Well, first you actually follow the root back to the trunk. I mean, it's not just digging down, but it's digging back to the trunk, because there's lots and lots of trees and lots of shrubs, and so you want to make sure that the root that you see is actually attached to the plant you think it is. And there have been many false reports of hosts among these parasites because somebody just pulled one up and said, oh, well, it's probably that shrub over there, without any knowledge of the root systems. Studying the underground structures of plants in general is very difficult because we can't see it the way we do the above ground parts. For seed germination studies on many of these parasites, they've actually developed pots with windows in the side, and they will put the seeds right next to the plexiglass so that they can see the seeds, and then they'll plant a bunch of hosts so that the hosts really get root bound in the pot, and they can actually see the roots as they come up to the parasites, and they can observe what's going on. That's the only way you can ever do direct observation on this stuff. You can't really do it outside of the soil environment. And in many cases, you don't have the microbes outside the soil environment that stimulate decay and growth and other things that happen below the surface, which is why we know a whole lot less about what's happening underground than above ground in plants and life in general.
Okay, let's thank him for a good talk.
Thank you.
For more information, please visit tlu.edu.